I would like to present Professor Steven Schwartzbach from University of Memphis, and he'll be talking about Plastid genome.
Well, I'm trying to share my, there we go.
Yes, we see it. It's just not in the full screen mode yet.
Okay, it should be coming up in a minute. My computer locks up sometimes. But what I'm going to talk about are two areas in Euglena that basically show Euglena does things differently. Okay, this first slide is 3D immunoelectron microscopy reconstruction of the Euglena cell, which was done by Tetsuo Safuno, a valued colleague and friend who is no longer with us. The importance is that here in yellow, we see the immunogold labeling of the chloroplast protein. It's not surprising that it's in the chloroplast. What was surprising is that it was in the Golgi apparatus. And this led to, at least in my lab, the biochemical studies to look at the processing. Okay, here what we've done is you look at the Euglena pre-sequence models, and they have a single peptide, what appears to be a transit peptide, and a hydrophobic membrane domain. But what we wanted to see in the data I will present is that these are all universally functional. In this case, what we've done is made a GFP fusion protein where the ER and Golgi are labeled with a dye that stains them red, and we see the GFP fluorescence in green. When we overlay the two, we very clearly see the perinuclear localization and yellow indicative of Golgi localization. The precursor is sent to the Golgi in mammalian cells. If we look at lysosomal labeling, we do not see it in the lysosome. So we clearly have a precursor that is Golgi targeted and apparently has a membranization signal on it as well. So now what we tried to do is analyze the different parts in vitro systems. So here what we've done is we've isolated Golgi membranes that have been post-labeled so that we could immunoprecipitate our CP2, and here's the membrane fraction. The Euglena precursors are polyproteins. We've shown that the protease that processes them is in the chloroplast. Therefore, if the precursor is processed, that indicates chloroplast localization. And what we find is that in the presence or absence of cytoplasm, we get mature protein processed that has been imported into the chloroplast, and for some reason we see a dimer that increases with time. Apparently, a portion is not fully processed, but that clearly shows that we go from the Golgi to the chloroplast. Okay, the next question we wanted to ask is this an obligate transport pathway, or can Euglena precursors be imported post-translationally from the cytoplasm? So what we've done is we've taken the full pre-sequence and a pre-sequence where we have removed the single peptide. So this is what would be presumably be presented within the Golgi transport facility, the translation products, and we find that both the complete pre-sequence as well as just the transit peptide does associate with the chloroplast. It is pelleted through Percol. However, if we treat with thermolysin, it is fully thermolysin sensitive. It has not been transported into the chloroplast. This happens in the presence of ATP and the absence of ATP. So we cannot post-translationally import into the chloroplast. We have obligate vesicle fusion. If we now look at the universal functionality of the transit peptide, looking for in vitro incorporation into peak chloroplasts, what we find is that with the transit peptide, it will associate in the absence of ATP, pyrice getting rid of the ATP with the chloroplast pelleting through percol. However, it is fully thermolysin sensitive. It has not been transported into the P chloroplast. However, if we add ATP, which is required for higher plant precursor transport, we find a thermolysin insensitive fraction. So this can mediate import into higher plant chloroplasts post-translationally, but not into Euglena chloroplast. Okay, so now this brings us to what I consider an enigma, someone being relatively ignorant of evolutionary biology. And to me, what stands out about this system is that the phylogenetically distinct Euglena plastids, which came from a green endosymbiont, and the dinoflagellate plastid, which came from a red endosymbiont, have 3 envelope membranes. They have the same type of pre-sequence containing single peptides, transit peptide, and stop transfer membrane anchoring sequence, and they have polyprotein precursors. These are not seen in any other system, to my knowledge. If we look at the 8 big complexins, which are presumed closely related to the dinoflagellates with their non-photosynthetic plastids, they do not share these precursor properties. And I guess to me the enigma is, how have these evolved? Now, it has been published that this is convergent evolution. When I look at this system, I find that hard to believe, simply because to independently develop this system, which is rather anomalous and does not appear to show any sort of selective features for where it came from, doesn't appear it would have happened twice. Okay, so to me, it's an enigma in my knowledge of evolutionary biology. Okay, the other thing I want to talk about is studies that have been done on bleaching, which again make Euglena unique. Bleaching is the permanent loss of the ability to form green colonies without an effect on cell growth or viability. And bleaching is caused by inhibitors of protein synthesis on 70S prokaryotic type ribosomes, such as streptomycin, indicative of requirement of chloroplast protein synthesis for chloroplast genome replication, and by inhibitors of prokaryotic chloroplast DNA gyrase, such as afloxacin. Okay, what we find when we look at the chloroplast genome during the process of bleaching, either afloxacin or streptomycin, which appears to be more effective, is 3 effects. We see a deletion, certain genes become undetectable. We see a lessening in the amount, but surprisingly, some show no appreciable change in distribution. Okay, this is approximately half of the genome. We see one rather anomalous finding. A large ribosomal protein, the gene for it is totally gone. What we find when we look at the rest of the genome is, more surprisingly, additional ribosomal proteins, tRNA genes, and most importantly, the ribosomal RNA genes are gone. Large subunit 5S RNA and small subunit ribosomal RNA are undetectable. Yet, we have found that certain parts of the genome are still maintained in these cells. Okay, this takes me to my second enigma, is that bleaching demonstrates that protein synthesis on chloroplast ribosomes is required for chloroplast genome replication, yet genes encoding the ribosomal RNAs, several small subunit and large subunit ribosomal proteins are undetectable after less than 60 generations. Certain parts, certain chloroplast genes are present at unchanged amount. How do these genes fragments replicate in the absence of protein synthesis on chloroplast ribosomes? Okay, finally, I'd like to thank the people who I've had the privilege of working with over my career in my own lab, in Europe, and again, my friend and colleague who is no longer with us, Tetsuo Sifunu, and his close associate, Tomo Ihara. Thank you, and I hope I made it in the time limit.